The overall goal of this procedure is to synthesize biofunctionalized Prussian blue nanoparticles for use as multimodal molecular imaging agents. This is accomplished by first synthesizing Prussian blue nanoparticles that contain either gadolinium or manganese ions, which enhance MRI signal. The second step is to attach fluorescent avidin onto the surface of the nanoparticles, which confers fluorescence imaging capability. Next, the avidin-coated nanoparticles are biofunctionalized with the desired cell-targeting biotinylated antibodies. The final step is to perform a quality control check on the biofunctionalized nanoparticles by measuring their size, zeta potential, and temporal stability. Ultimately, the biofunctionalized nanoparticles are utilized in multimodal molecular imaging applications to visualize a specific targeted population of cells within a mixture using fluorescence imaging and MRI.
Current commercial imaging agents are typically single-mode and offer resolution only at the anatomical level. Prussian blue nanoparticles combines two complementary imaging modes, MRI and fluorescence, and allow for molecular imaging at subcellular levels.
The applications of multimodal nanoparticles extend toward diagnosis of cancer and inflammatory diseases, in addition to monitoring of their treatment responses. This is because biofunctionalized nanoparticles can target and bind to a population of cells within a specified region of the body.
Although this method can provide insight into cancer and inflammatory disease diagnosis and response to treatment, it can also be applied to other pathological conditions